Cellular respiration. Cellular respiration consisted of glycolysis and Kreb steps. Second, Kreb cycle or called as citric acid cycle. Third, electron transport chain or called as ETC. Glycolysis. Glycolysis is the breakdown of glucose by enzymes releasing energy and pyruvic acid. Steps. Step 1. Enzyme hexokinase. It is an enzyme that catalyzes the phosphorylation of many six-membered glucose-like ring structures into glucose 6-phosphates. This is the reaction: glucose plus ATP through enzyme hexokinase producing glucose 6-phosphate plus ADP and plus hydrogen. Step 2. By enzyme phosphoglucose isomerase. It is an enzyme that catalyzes the conversion of glucose 6-phosphate to fructose 6-phosphate (G6P) to F6P by isomerization reaction. And this is the reaction: G6P producing the fructose 6-phosphate F6P through enzyme phosphoglucose isomerase. Now we're moving to step 3. Phosphofructokinase. It is an enzyme that catalyzes the reaction of fructose 6-phosphate into fructose 1,6-biphosphate or FBP. FBP is splitted into two kinds of 3-carbon molecules that are isomers of each other, which are DHAP, dihydroacetone phosphate, and PGAL, glycerol dihydrophosphate. Eventually, DHAP is converted into PGAL, a more useful molecule for our purposes, through enzyme aldolase. Now, we're moving to glycolysis 2. It is about how PGAL is converted into pyruvate. After still more chemical reactions, the PGAL is going into pyruvate through the process such as redox and phosphorylation, dephosphorylation, isomerization, dehydration, and dephosphorylation again. In these processes, after each PGAL contributes to the reduction of two molecules of NAD+, each of the two 3-carbon molecules receive another phosphate from inorganic phosphates. The phosphates received are then removed, forming two ATP molecules. Shortly thereafter, the remaining phosphate groups are then removed to form two more ATP molecules. The end molecules are two 3-carbon pyruvate ions. As we progress, we will updating a chart that looks like this. So far, we made 4 ATP and spent 2. Then we recharge 2 molecules of NADH and 0 FADH2. Remember that there are 2 pyruvates produced from each glucose. Thus, as in glycolysis 2, both the intermediate step and the cycle occur twice. Prep steps occur in cytoplasm just like glycolysis. The 3-carbon molecule of pyruvate are modified using 2 coenzyme A. Coenzymes store energy in their bond and help an enzyme work more effectively. The enzyme pyruvate decarboxylase catalyzes this complex reaction. The first event is the decarboxylation, the removal of carboxyl group to form carbon dioxide. The intermediate compound then donates 2 electrons to an NAD positive to reduce it to NAD negative. Then an hydrogen ion is donated by coenzyme A to complete the formation of NAD+, NADH, forming 2 reloaded molecules of NADH. The coenzyme A ion then attaches to the positive charge left on the acetyl ion, forming acetyl-CoA. At the end of the intermediate steps, two acetyl coenzyme A and two carbon dioxide generated along with the two reloaded molecules of NADH. So here we made 0 ATP, 2 NADH, and 0 FADH2. Krebs cycle. It is defined as the sequence of reactions by which most living cells generate energy during the process of aerobic respiration, and it is also known as citric acid cycle, that takes place in the inner compartment of mitochondria, known as the matrix. Every single steps are performed by enzymes. So this is the Krebs cycle. First, we draw the arrow first, and then we write the front letters of each of the citric acid. Acetyl-CoA have two carbons. The acetyl-CoA from the prep step adds on to a new form of carbon molecule called oxaloacetate to form an intermediate molecule. 2 carbons plus 4 carbons equals 6 carbons. From here we add a series of enzymes to convert each molecule into the next one. The next molecule is isocitrate, which is similar to citrate and also has 6 carbons. Next, alpha-ketoglutarate is formed, which only has 5 carbons. So what happens next to that 6 carbon? It will release in the form of carbon dioxide. Next up is a molecule called succinyl-CoA, which has 4 carbons. Based on the last step, you can probably guess what happened to that 5th carbon. It was released in the form of a second molecule of carbon dioxide. Next up is a molecule called succinate, which also has 4 carbons. Then comes fumarate, which has 4 carbons, and then malate, which too has 4 carbons. We also add a molecule of water here to get this final molecule. Last, malate is converted back into oxaloacetate, the place where we started, and the cycle begins for the second time. If you are forced to memorize the Krebs cycle, here is a little trick that can be used. CIKSSFMO. Cindy is keto, so she fits more obnoxiously. So now we map up how the Krebs cycle cycles carbon and where the carbon dioxide waste product comes from. Let us go back in and add in ATP and also add in all of our electron carriers. We reload 3 molecules of NADH during the Krebs cycle. The first when we convert isocitrate into alpha-ketoglutarate, and the second when we convert alpha-ketoglutarate into succinyl-CoA. The third is when we convert malate into oxaloacetate. We recharge a single molecule of ATP between succinyl-CoA and succinate, and then we also reload a new molecule of FAD into FADH2 between succinate and fumarate. However, all of the above is per molecule of pyruvate, and since each glucose is broken into 2 molecules of pyruvate, this cycle can turn twice per molecule of glucose. One rotation of this cycle makes 1 ATP, 3 NADH, 1 FADH2, and 1 carbon dioxide. But since the cycle turns twice, we actually make double for each molecule of glucose we put in. In total, we now have 4 ATP, 10 NADH, and 2 FADH2. Electron transport chain, or called as ETC, or even called as chemiosmosis. The electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria where the redox reaction occurs to form a gradient of protons that drives the creation of adenosine triphosphate (ATP) via chemiosmosis, and the process involved is oxidative phosphorylation. This is the structure of mitochondria. The most outer part is called as outer membrane mitochondria or OMM. The inner membrane mitochondria or IMM. The space between the inner membrane mitochondria and the outer membrane is called as intermembrane space or IMS. The electron transport chain usually occurs in the IMM, inner membrane mitochondria, and the most inner space is named as matrix. Inner mitochondria membrane contains 4 sets of enzyme complexes, which name from 1 to 4 in Roman numerals. The matrix in mitochondria also contains numerous other cycles like Kreb cycle and beta oxidation. This cycle producing reducing equivalents NADH and FADH2. These reducing equivalents contain electrons which are donated to the set of enzyme complexes, which is complex 2. The electrons then travel from first to the fourth enzyme complex, and in this movement, energy is generated. This energy is utilized in pumping hydrogen ions into the intermembrane space from the matrix of the mitochondria. This continuous pumping of hydrogen ions into the intermembrane space causes the generation of higher concentration of hydrogen ions in the intermembrane space as compared to the matrix of the mitochondria. This generates a positive charge in the intermembrane space and a negative charge in the matrix of the mitochondria. This is called the electrochemical gradient. The hydrogen ions cannot cross against this electrochemical gradient because the inner mitochondrial membrane is non-permeable to ions. So here we require a special transporter. The enzyme ATP synthase comes into play. The ATP synthase transports hydrogen ions in the matrix of the mitochondria and uses the energy generated from the flow of hydrogen ions to phosphorylate adenosine phosphate into adenosine triphosphate. This is the basis of generation of energy in the form of ATP, which is a currency of energy in our body. In addition to all 4 complexes, we also have 2 extra prosthetic groups called the coenzyme Q or called as CoQ and site C or called as cytochrome C. Complex 1 of ETC is named as NADH dehydrogenase complex or NADH oxidoreductase. The first complex is the L-shaped protein complex, which is present in the inner mitochondrial membrane IMM. It has 2 arms. The vertical arm is located inside the IMM, whereas the horizontal arm is located inside the matrix of mitochondria. Complex 1 is called as NADH dehydrogenase complex because it dehydrogenases which removes hydrogen. It removes hydrogen from the reduced form of NADH and it is called a complex because it is also contains FMN, flavin mononucleotide, and iron sulfur, FES, of compounds. It is also called as oxidoreductase because dehydrogenation is an example of oxidation-reduction reaction. The main job of this complex is to receive electrons from NADH and transport it to further to ETC. Complex 2 is also called as succinate dehydrogenase complex. It is called as dehydrogenase because it removes hydrogen from compounds. In this case of succinate, it will remove hydrogen from the succinate and oxidize it into fumarate, which is a step of Krebs cycle. Similarly, it is also called as a complex because it also contains iron-sulfur clusters (FeS) in addition to succinate dehydrogenase. This second complex is a part of Krebs cycle, which catalyzes the reaction for oxidation of succinate to fumarate. In this reaction, the reducing equivalent FADH2 is produced, which is utilized for donation of electrons in the electron transport chain. The complex tree of the ETC is called the cytochrome reductase. It is also known as the Q-cytochrome C oxidoreductase. Sorry, I'm being typo. It actually have letter Q in front of the cytochrome C oxidoreductase. Cytochromes is a group of proteins which have heme as their complexes or the prosthetic groups. They also have iron core, in which the ion can exist in an oxidized or reduced form, depending on the electrons it has. The complex tree contains three types of cytochrome, namely cytochrome B, cytochrome C1, and cytochrome C. The main job of these complexes is to accept electrons from the ETC and then transport it to the cytochrome C. The cytochrome C then transport these electrons to the complex 4 of the ETC. The complex 4 is called as cytochrome C oxidase. It is a heme and copper containing complex whose main job is to oxidize cytochrome C, that is, to receive electrons from cytochrome C, and then it uses those electrons for the reduction of oxygen to water. Hence, it is called as cytochrome C oxidase. Now, we're going to part 2 of ETC. It is about how ETC works. But first, we need to know very well on part 1 ETC, second the Krebs cycle, glycolysis, and the overview. Here is the illustration which illustrates the membrane structure of mitochondria as well as the organization of the various electron gene complexes. When we compare it to the structure of mitochondria, we can see that here is the four mitochondrial membrane, such as outer mitochondrial membrane, intermembrane space, inner mitochondrial membrane, and the matrix. In the matrix of mitochondria, we have biochemical cycles such as Krebs cycle as well as beta oxidation of fatty acids. These biochemical cycles generate reducing occurrence called NADH and the FADH2. The electron transport begins when the NADH donates its electrons to the first complex. In the first complex, the electrons first received by the flavin mononucleotide prosthetic groups (FMN). The FMN then donates the electrons to the FeS iron-sulfur proteins. These are redox reactions in which the iron ions go from the ferrous state to the ferric state, depending upon when they receive. and when they donate the electrons. In the next step, the FES protein donates electrons to CoQ, coenzyme Q. Q is the ubiquinol, which is a freely permeable molecule inside the inner mitochondrial membrane. Ubiquinol is the reduced active antioxidant form of coenzyme Q. These redox reactions generate energy, which is used to pump 4 hydrogen ions from the matrix of the mitochondria into the intermembrane space. The coenzyme Q then combines with 2 hydrogen ions from the matrix of the mitochondria and gets converted to the reduced form of ubiquinone. In complex 2, also known as succinate dehydrogenase complex, it contains the enzyme succinate dehydrogenase. This enzyme catalyzes the conversion of succinate fumarate in the Kreb cycle. This is oxidation reaction which causes the generation of FADH2. The FADH2 stay inside the second complex and donate the electrons to the FES of complex 2. The electrons are then transferred to the ubiquinone in a similar way as the electrons are transferred from the first complex. The ubiquinol is a freely permeable molecule inside the inner mitochondrial membrane, which is used to transfer electrons to the third complex. In the third complex, the electrons are first received by the cytochrome C1. The cytochrome C1 then transfers the electrons to FES proteins, which then donate the electrons to cytochrome B. These are all redox reactions. The electrons are then transferred to the cytochrome C, which is an intermediate protein used to donate electrons to the fourth complex. The redox reactions inside the third complex cause the pumping of 4 hydrogen ions to the intermembrane space. The cytochrome C underneath the electron of fourth complex. In this complex, the electrons are first received by the copper ions, which are A-type. The copper ions then donate the electron to the cytochrome A. The cytochrome A transfers the electrons to copper ions, which are of a B-type. These copper ions transfer the electrons to cytochrome A3. The cytochrome A3 in the final step donates the electrons to the final electron acceptor, which is the oxygen. The oxygen combines with 2 hydrogen ions from the matrix S of mitochondria and get reduced to water. This redox reaction in the fourth complex causes the pumping of 2 hydrogen ions to the intermembrane space. All this pumping of hydrogen ions into the intermembrane space causes a generation of higher concentration of hydrogen ions into the intermembrane space, as compared to the low concentration inside the matrix. Inner mitochondrial membrane is an impermeable membrane to ions, so hydrogen ions cannot cross the membrane as such. ATP synthase is present inside the inner mitochondrial membrane, and it uses the flow of hydrogen ions from higher to lower concentration to generate energy. This energy is used to phosphorylate ADP into ATP. This is the basis of generation of energy inside the body. The working of ATP synthase can be compared to the working of hydroelectric power project. The water that is collected behind the dam can be compared to the hydrogen ions in a higher concentration in the intermembrane space. The turbines of the dam can be compared to the enzyme of ATP synthase. Now, Let us talk about the overall energy calculations per molecule of glucose that passes through the biochemical cycles of glycolysis, Kreb cycles, and the electron transport chain. In glycolysis, when glucose is converted to pyruvate, we get a net of 2 ATPs and 2 NADH. In the next step, when pyruvate is converted to acetyl-CoA, we get 2 NADH. In the Kreb cycle, when glucose passes through one cycle of Kreb cycle, it gives 2 GTPs, which are energy equivalent to 2 ATPs. We also get 6 NADH and 2 FADH2. All these reducing equivalents are then metabolized in the electron transport chain. In the electron transport chain, we metabolize the 2-NADH from the conversion of glucose to pyruvate. Also, the 2-NADH from the conversion of pyruvate to acetyl-CoA, and both 6-NADH and 2-FADH2 from the Kreb cycle. In the electron transport chain, for every 4 hydrogen ions that flow through the ATP synthase, 1 molecule of ATP is generated. So, when 4 hydrogen ions pass through the ATP synthase, it is utilized for the phosphorylation of 1 molecule of adenosine diphosphate. A 1 molecule of NADH, which donates its electron to the electron transport chain, causes the movement of 10 hydrogen ions from the matrix to the intermembrane space. These hydrogen ions then flow back through the ATP synthase, give rise to 2.5 ATPs. In a similar way, 1FADH2 causes the movement of 6 hydrogen ions to the intermembrane space, which then move back, give rise to 1.5 ATPs. Sorry, I have written there 15 ATPs for 1FADH2 equal to 6 hydrogen ions. It should be 1.5 ATPs. Using these values, we can calculate that 2 NADH from the conversion of glucose to pyruvate will give 5 ATPs. Similarly, conversion of pyruvate to acetyl-CoA will give 5 ATPs in the electron transport chain. 6 NADH from the Kreb cycle will give 15 ATPs, and 2 FADH2 will give 3 ATPs. In a total, we get 20 ATPs from the Kreb cycle. We get 7 ATPs from the conversion of glucose to pyruvate in glycolysis. We get 5 ATPs from the conversion of pyruvate to acetyl-CoA. Overall, we have 32 ATPs per molecule of glucose, which passes through all these biochemical cycles.